Welcome to part 2 of the Central Dogma series. This video will review the importance of DNA and DNA replication. In the previous videos, we talked about the building blocks of genes, DNA and RNA, what they are made up of, and how they are built. DNA stands for deoxyribonucleic acid and contains and stores all genetic material, or genes, in all living organisms. The structure of DNA is a twisted double helix. If we untwist the DNA, it looks like a ladder. DNA is made up of 4 nucleotides: adenine, thymine, guanine, and cytosine. Each pair of nucleotides makes up a rung of DNA. To construct the DNA structure that looks like a ladder, adenine and thymine always pair with each other with 2 hydrogen bonds. Cytosine and guanine always pair with each other with 3 hydrogen bonds. Remember A to T and G to C. Attached to each nucleotide, there is a sugar and a phosphate group. The outer structure of DNA is connected by phosphodiester bonds between a sugar and a phosphate group of the neighboring nucleotide. With these bonds, we can add more and make a larger DNA strand. We've built a small strand of DNA. The 5' end is designated by the phosphate group, and the 3' end is designated by the sugar group. Remember, the DNA strands are anti-parallel, meaning they run in opposite directions. If we take cells from different parts of this tomato plant, like the flower, tomato fruit, leaf, and stem, and look inside the nucleus, we will find all cells contain DNA, and the DNA is nearly identical. But the expression of certain genes is what makes these plant parts different. How are there so many copies of DNA since every cell of the plant has DNA? DNA undergoes DNA replication, the process of copying DNA. How does DNA replicate? We make copies of things with a copy machine. Plants have their own version of this. Let's talk about how it works. Replication occurs during interphase. An interphase consists of 3 important phases: cell growth, DNA synthesis, and finally, additional growth and preparation for mitosis. DNA replication occurs during the DNA synthesis phase, or S phase. Mitosis occurs within the nucleus, followed by cytokinesis, where one cell divides into 2. Prokaryotes, like bacteria cells, don't have a nucleus, but still have DNA replication, so their progeny cells have the information they need to live. In a eukaryotic cell, like a plant cell, DNA replication occurs in the nucleus. The mechanism for DNA replication is the same for prokaryotes and eukaryotes, but how and where the process occurs is slightly different. The tools needed for DNA replication include DNA and an assortment of enzymes with specific tasks. Our first enzyme is helicase, which unzips the DNA. Primase primes the initiation point. Helicase and primase together form a primosome, unzipping and priming at the same time. DNA polymerase builds the complementary strand of DNA. And lastly, ligase fills in the gaps within the lagging strand. Let's talk about the process occurring in prokaryotes first. The double-stranded DNA is organized into a region in the center of the cell called the nucleoid, which is in the cytoplasm of prokaryotes. It is circular and small. It has one point of origin to begin replication. There are 2 chromosomes that are unzipping and priming the DNA in both directions. This allows DNA polymerase to build 2 new strands simultaneously. After the DNA polymerase has copied the original strand, we are left with 2 identical copies of DNA. DNA replication happens at the same time the cell elongates and divides. When DNA replication is complete, there is 2 identical cells. In eukaryote replication, the process is slightly different. The DNA is replicated before the cell divides and has multiple points of origin to begin replication. Eukaryotic DNA has multiple points of origin throughout the strand. In the end, this still leaves us with 2 identical strands of DNA. Remember, DNA replication occurs during the DNA synthesis, or S phase, of the cell cycle. Mitosis occurs within the nucleus, followed by cytokinesis, where one cell divides into two. Let's review the four main enzymes required for DNA synthesis: helicase, primase, DNA polymerase, and ligase. Helicase is responsible for opening or unzipping the DNA strand. Primase primes the original strand to prepare for DNA replication. The areas where primase has primed are indicated by the yellow portions on the figure. DNA polymerase finds the primed area and attaches to the strand where it can begin building the new DNA. There are multiple segments of the lagging strand of the DNA called Okazaki fragments. This is because DNA replication occurs from the 5' end to the 3' end. The leading strand has no fragments because new nucleotides are added to the 3' end as soon as helicase opens up the DNA at the replication fork. Ligase glues the Okazaki fragments together, as indicated by the blue portions, to complete the lagging strand. All the enzymes work together nearly simultaneously to replicate the DNA strand. Let's review what we have learned. DNA replication differs between prokaryotes and eukaryotes. Prokaryotes have one point of origin to begin replication and divide into two new cells while the DNA is being duplicated. Eukaryotes, however, have multiple points of origin throughout the DNA strand, and this is then followed by cell division or mitosis. DNA is the important genetic code within plants and all living things, and is the first step of central dogma, which is the process of DNA code being read into functional products.